Vanakkam. Welcome to this course on classics in neuroscience. In the last weeks, we looked at neural circuitry. Specifically, we looked at neural circuitry that involved specific forms of perception. We also looked at cortical organization, cortical columns and cortical processing. In this week, starting with this video onwards, we will look at neural systems and the neural basis of behavior. So, what is shown here are pathways or a system that is responsible for a certain type of behavior. In contrast to the previous weeks where we looked at specific circuitry that underlie specific form of perception. In this week, we will look at more global systems that are responsible for behavior. That is, when I say global systems, I am looking at a network of neural circuitry, not just one circuit. So, widely distributed systems that underlie and mediate behavior. Also, in the 1950s, there was evidence that suggested the brain is also a gland. So, there will also be a discussion that considers the brain as a gland in these set of videos, and the nature and the role of the reticular activation system in arousal will be discussed. We will also discuss about the nature of sleep, how after the discovery of rapid eye movements, it was postulated, it was hypothesized that sleep is not a completely passive state, but rather an active state of behavior. Also, there was the introduction of operant conditioning as a crucial critical paradigm for studying behavior. Then the two-state theory of feeding will be discussed. Then we discuss the nature of the pituitary gland as the master gland, and the neural control of the pituitary is also something that we will discuss. In this video, we will look at the reticular activating system and arousal. In the last weeks, we looked at specific neural circuitry that mediate specific types of perception. For example, visual perception, for example, proprioception. We looked at those. But then there are also global phenomena that underlie what constitutes global behavior or the behavioral state of the organism. That is, pathways and circuits that mediate specific types of perception and behavior. What are these types of perception? Visual, proprioception. For example, we looked at the muscle spindles and the 1A afferents and the Golgi tendon organs. Then we also briefly discussed touch receptors. Although we did not discuss in great detail, there are also these olfactory receptors and auditory receptors. But in contrast, there are also systems, not circuitry. Systems means a network, several circuits that communicate with each other. So, systems that have more global effects, that is larger phenomenon, behavior itself. Also, some of these are responsible for releasing or responding to circulating hormones, chemicals. So, this also in some way sets the behavioral state of the organism. Sherrington, in one of his landmark papers and also in his book, describes some of his observations in the cat. So, he performed experiments in which there was transaction at different levels, and he observed activity at the level of the spinal cord below the level of the transaction. If there is low transaction of the brain stem that happens, then what happens is this leads to a form of paralysis that is dominated by flaccidity, flaccid paralysis. So, that is looseness. So, this is almost at the level of the spinal cord, because this is a low transaction that is happening at the level of the brainstem. So, the symptoms are dominated by flaccidity. But if the transaction happens above the level of the red nucleus, specifically between the superior and the inferior colliculus. So, there is this body containing 4 nuclei, corpora, quadrigemina. Between the superior 2 bodies and the inferior 2 bodies, between the superior colliculus and the inferior colliculus, if the transaction is done, this leads to a state of very high activity in the muscles, so much that the animal can support its own body weight. The entire cerebral cortex is transected, but still the animal can remain strong enough, or the activity is strong enough to keep the animal on its own limb, it can support its own body weight. So, essentially a state of heightened extensor tone. This is called as decerebrate rigidity, decerebrate rigidity. A unique case. This is observed only when the transaction is at the levels that I described, above the red nucleus, between the superior and the inferior colliculus. This he reported in a paper, but also in his book on the integrative action of the nervous system. Then in the 1930s, Frederick Bremer performed experiments that involved transactions, low transactions and high transactions, and observed the state of the brain above the level of the transaction. What is the difference between the Sherrington studies and these studies? Sherrington studied the activity below the level of the transaction. Bremer studied the activation levels above the level of the transaction. Okay. So, if the transaction is low, it is called as low transaction, he also called as encephalae isolae or isolated encephalon. This had no effect on normal walking or sleeping patterns. However, if the transaction happens above the red nucleus or isolated brain, this produced a form of permanent deep sleep. These observations pointed to the midbrain pons region, pontine region it is mentioned here, that is that nucleus called pons, as a possible site of that mediates mechanisms of arousal. This is the inference that we could come up with after observing Bremer's results. In the 1940s, Robert Morrison performed experiments where he electrically stimulated the thalamic regions. Remember, the thalamus is the relay center of the brain. Many systems, many nuclei relay their neurons or relay the information by sending neurons or axons to the thalamus. So, it is the relay center. So, what happens if we activate, electrically stimulate the thalamus, and if we study the response of the cerebral cortex? So, if the stimulation was on the sensory relay nuclei, then the response in the cerebral cortex was limited to the cortical projection areas. So, wherever this relay nucleus is projecting, to those areas there was activation. So, you activate the relay nucleus, and wherever these relay nuclei are projecting, in those areas you find activation in the cerebral cortex. But if they activated the interposed nuclei, this led to a relatively broad activation, what is called as widespread activation, that increased with repetitive stimulation. So, if you activate interposed nuclei, there is a wider activation of the cortex. This led to the inference that maybe these interposed nuclei are non-specific thalamic nuclei, and their responses may be considered to be recruiting responses. In comparison, relay nuclei essentially activate only specific regions. So, perhaps these are more specific responses. So, fundamental difference between the two. Later, in the late 1940s, Giuseppe Moruzzi of University of Pisa in Italy and Horace Magone of University of California, Los Angeles, they performed high frequency stimulation in the brain stem, and they observed activity in the cortex. So, essentially they found that this high frequency stimulation produces an arousal in the cerebral cortex, and they found a link between the brainstem system and the thalamic system. And they also found that the sites where the stimulation happened in the brainstem correlate with not with a specific set of cells, but rather with many cells from the medulla to the diencephalon. So, rather widespread activation. This region where they stimulated came to be called as the reticular formation, also called as the reticular activation system. Lesions of this reticular formation produce deep sleep and practically blocks cortical arousal. So, you look at these. So, this is where the red nucleus is, and this is where the thalamus is. Of course, also shown here are specific nuclei that form the basal ganglia, such as caudate nucleus, globus pallidus. So, this is the location that we are speaking about. So, if there is a sensory stimulation, it activates the specific sensory pathway, as we have previously seen, that leads to perception, but it also activates through the RAS, through the reticular activating or the reticular activation system in the brainstem. It also activates a non-specific pathway that leads to cortical arousal and then leads to what is called as conscious perception. So, this is the phenomenon of arousal, attention and consciousness. Understanding this is critical to management of patients who are sleeping for a long time, that is coma, patients in coma, and to assess whether a patient is brain dead, assessment of brain death. Yes. Only in the late 90s, the neural basis of consciousness has been studied as a discipline in its own, studied as a subject in its own. Of course, people have been interested in studying consciousness at the level of the cortex. For example, Francis Crick, Christoph Koch and some others have been interested. Currently, there is interest in studying consciousness with more specificity, studying specific aspects of conscious movement and conscious perception. So, with this we come to the end of this video. In this video we looked at arousal and the reticular activating system and how activation of the reticular activation system leads to non-specific activation of the cortex, leading further to what is called as conscious perception. Thank you for your attention.